last but not least, Chris is going to give us a bonus talk, and then we'll move on to 15 minutes of questions, and then we'll have a wrap up of 15 minutes. So, Chris.
Thank you very much. Thank you, Martin, and thank you for creating this slot for me and asking me to present in it. And I apologise. It's another talk about factor H binding protein, or at least part of it is. Just to go back to a slide I showed earlier on, in terms of the research that we've been doing in the lab, we have been interested in acquisition of nutrients, particularly carbon and energy sources, and in the avoidance of immune killing. And in respect to avoidance of immune killing, it's just worth taking a step back and looking at human genetics and variation in humans that predispose individuals to meningococcal disease. And this is just a schematic of the complement system in humans. And outlined in blue are all the factors in complement which there are polymorphisms or deletions in, which render people highly susceptible to meningococcal disease. So it shows that complements are really important in terms of protecting us from this bacterium. Just a couple of things to point out. The serum bactericidal activity, which has been mentioned by several people during this particular session, is based on the lytic ability of bacteria to induce the terminal pathway of complement, which leads to assembly of membrane attack complex and insertion into the membrane and destruction of the bacterium. And the activation pathways based on antibodies and on lectins are regulated by the alternative pathway, which has got major negative regulators, particularly factor H. And this is where factor H binding protein comes into the story. So we worked a number of years ago to start looking at the ability of factor H to bind the meningococcus. This is work we did with Bob Sim here at the complement unit, with Ian Fevers as well, old collaboration. And we showed that the bacterium can bind factor H, different strains bind factor H via proteins of different sizes. So we knew at that time the factor H receptor on the bacterium was polymorphic, and the presence of factor H in the added to serum protected meningococcus from being killed. It was Dan Granoff who showed that the factor on the bacterial surface which bound factor H was factor H binding protein. There was an amazing convergence of virulence and immune evasion together with the vaccine work which was being done at Pfizer and GSK Novartis at the time, that this turned out to be the same thing. The factor H binding protein was the antigen or key antigen for GSK and the sole antigen for Pfizer. And we solved the crystal structure of factor H binding protein with two barrel protein on the bottom with the complex with factor H in blue on the top. And those are two domains of factor H, the SCR 6 and 7 of a protein, which has got 20 SCR domains. These are short regulatory sequences of about 60 amino acids each. So without data in hand and vaccine development really pointing at factor H binding protein, this led us to consider two things. First of all, can we make non-functional factor H binding proteins? Well, to do that, we knew the interface between factor H binding protein and factor H, and so we were able to mutate factor H binding protein in that interface, replacing all amino acids with alanines to get rid of the R side groups. And we did this for variant 1, variant 2, variant 3 factor H binding proteins, or B and A subfamilies. And we have a sort of complete catalog of sites marked in red where you mutate the amino acid to an alanine and you get less than 10% binding of factor H. So this is an area for development of vaccines because the current vaccines have not inactivated factor H binding. So there is some evidence that they may not bind factor H in the formulation or as fusion proteins. It's not certain. The other issue is whether you want more than one protein to give you more coverage or give you more depth of coverage. And we considered this, and with a structure in hand, we decided to use factor H as a factor H binding protein as a molecular scaffold. So this is a soluble 2 beta barrel protein, which has been described before, is tethered to membrane by a lipid anchor. Clearly, there are beta barrel proteins which are highly immunogenic. 4A, which has been worked on by many people for many years, central protein immunogen in OMV vaccines, which are embedded in the membrane. These are difficult to express because they are lipophilic in nature, hydrophobic in nature. They have to exist in the hydrophobic environment, in the outer membrane. But there are known surface loops of Por A, which are highly immunogenic, in particular the VR2 loop, which has been shown to be the sequence which is most recognized when, or important for immune responses when OMV vaccines confer strain-specific immunity. So what we decided to do was to take our soluble beta-baryl protein and decorate it with the loops which are expressed from an insoluble beta-baryl protein in the membrane onto a factor H binding protein. So here you can see the different sites that we put the loops, which are these sort of colored balls, on top of the gray beta-baryls. And you can see that for some of these loop additions, we get no change in the binding of factor H to factor H binding protein. The binding is incredibly strong. This is dissociation constants in the nanomolar range. But if we put the loop in the position 183, then this abuts into the factor H binding site, and we get no binding detected. So here, within a single step, we can make a multivalent vaccine, and we can make it a non-functional factor H binding protein, so this will not bind factor H. So how we use this for the genome library? Well, you've heard a lot about how we can exploit the genome library and genetic sequences for choice of antigens and to understand coverage. And so this is a version of a tree from on the left-hand side of factor H binding protein sequences done by Deal using the MGL library sequences. And from that, we can choose judiciously different factor H binding proteins to cover the genetic diversity. We can superimpose on that the frequency of those particular alleles in the meningococcal population, so that, again, we're choosing alleles which should cover commonly expressed factor H binding proteins. And then on top of that, we can look in the genome library to see what Por A's are expressed by strains which either have the same, have a homologous exact sequence of the factor H binding protein, or have a different factor H binding protein. That's the data on the right-hand side, where we can see the common Por A VR2 loops in blue, showing the sequences which are present in strains with a factor H binding protein in the factor H binding proteins that we've selected for vaccine development, or in red, those Por A VR2 sequences which are not in a strain which we'd expect to be covered by factor H binding protein. So we can then mix and match our factor H binding proteins and Por A's. We're doing this in collaboration with Mark LaForce at the Serum Institute of India to produce a polyvalent 4 FHBP, 4 Por A loop vaccine, specifically designed for meningococcus B, specifically designed for eradication of MenB, or sorry, control of MenB, with a public health remit of thinking if the pentavalent vaccine in sub-Saharan Africa works and we then get outbreaks of MenB in sub-Saharan Africa, what can we do about it? And that's what we've got in mind here. So we're using the genomic sequences at the moment, clearly with the sequences which are available, but as the library becomes global, we hope this is sort of, we can use this information to do a sort of pick and mix type of vaccine for low income countries. So that's what I want to say about vaccine development. I'm going to talk then a bit about host pathogen interactions. And just to go back to the paper that was published, this was the first paper, I think, really describing the genome library, collaboration between the group here, Ray J in Manchester, and Julian at the Tang Center, led by Dot. And it shows clearly the population structure of meningococcal disease in England and Wales. And if you were to superimpose the population structure of carriage isolates, it would look very different. There may be some overlap. You'll see some of these clonal complexes present in the carriage populations, but there'll be other clonal complexes. So when we were talking about the sort of shift between commensal and pathogen behavior that was mentioned earlier on, we're quite keen to avoid the sorts of issues of comparing without recourse to looking at the population structure. And I just show this piece of data from a paper that we published a number of years ago with Martin Ray, with the Deal's help. Holly was involved as well in this. This was looking at a specific polymorphism in the capsule biosynthesis locus. It's the insertion sequence that David mentions, IS1301, can often be found coming into the capsule biosynthesis locus. And we found a couple of years beforehand that when this insertion sequence goes upstream of the capsule biosynthesis and upstream of the capsule export operons, then it can lead to upregulation of capsule expression and resistance to complement killing. So we're really interested to see whether this polymorphism was associated with disease or carriage. And this was our first, this is figure 1 from the paper, where things look really interesting. So we look at different serogroups. This polymorphism is more present in disease carriages, disease isolates versus carriage. And if you look across all of the serogroups, then it's a highly significant association with this insertion sequence. But as soon as we broke this analysis down into different clone complexes, this association just disappeared in front of us. And it just highlights the problems of comparing different clone complexes. You're essentially comparing apples with oranges. I think that was a salutary lesson to us. And we've done the same analysis that the Deal more recently has done the same analysis with more polymorphisms that we've found to increase capsule expression, and we don't see an association with any of the polymorphisms, all the polymorphisms put together when we control for clonal complex. And that sort of led us towards the recent work, which I'm just going to talk about very, very briefly, which is undertaking a bacterial genome-wide association study. And this is done with Martin and Danny Wilson and Sarah, who is in Danny's group. And she essentially, Sarah, looked at the presence of polymorphisms, looking at doing a KMA analysis, 30 base pair windows across the genome, initially doing a discovery study in clone complex 11 strains, looking at comparing invasive cases, so isolates from invasive cases from those with carriage. That showed that there's a polygenic nature for meningococcal disease. I think it's something we realized. Many of our favorite antigens are up there, including capsule biosynthesis, TSBAs up there, some genes involved in LPS biosynthesis, and gene FBA, which Marco and Isabel mentioned, which is upstream of factor H binding proteins. We got very excited by that. There were some synonymous SNPs in FBA, which we thought might add an extra FNR binding site, but it didn't seem to affect expression. But once we performed a replication study in clonal complex 41, 44, factor H binding protein, FBA locus really came up as the strongest set of polymorphisms. And actually, I haven't included the slide, but we have found functional polymorphisms in some of the associated with some of the SNPs, and those seem to be affecting the structure of the RNA thermosensor in factor H binding protein. That data was validated by experimentally by looking at RNA structures by Gabriele Virani in the University of Washington. So that's really as much as I wanted to say here. I want to say thank you to people, and I want to say thank you to the MRF who funded my group. I think one of the first grants I ever had was from the MRF, and it funded Rachel, who's been in my group for many, many years. And I was extremely proud to be part of the MRF as on the panel and to be a trustee for a number of years. We'd also want to say thank you to Haley and Maria, who are involved in the GWAS study from our side, and Sarah. I've got Haley twice, there's only once on the picture with her, Sarah Hollingshead, who has left the group now, but was instrumental in designing the chimeric vaccines, which was initially supported by Action Medical Research. And I said, it's a continuing collaboration with Serum Institute of India. Thank you.